Welcome to tonight's virtual CJD Foundation conference session with Professor Simon Mead. If you have questions during the program, you can type them into the box to the right of your screen, and we'll review as many as we can after the presentation. We hope you'll join us for the upcoming virtual conference sessions, which will be listed on our website. And now, Dr. Brian Appleby will introduce our speaker.
It's my pleasure to introduce Dr. Simon Mead. Dr. Mead is a neurologist who works at University College London in Queen's Square in the UK. He is a prion disease researcher, and although he may not refer to himself as a geneticist, one of his areas of expertise is genetic aspects of progressive brain diseases, very specifically prion disease, as well as things like frontotemporal dementia. And we're very fortunate to have Simon. He's a clinician as well as a researcher, and I've heard him speak in the past to family members, and he does an excellent job of distilling something that's very complicated to something that's very understandable. So thank you very much, Simon, for joining us today.
Thank you very much for the kind invitation to present the CJD Foundation webinar. As introduced, I'm a neurologist that works in London at the National Prion Clinic, and I also have a research group at the MRC Prion unit at UCL that looks at genetic factors, both in inherited prion disease and sporadic CJD. And I'd love to tell you about this work this evening. So I appreciate that there's probably quite a mixed audience here. Some of you may be old hands and have attended many webinars on the subject over 10 years or more, and others, this disease will be all too fresh for you. So I hope I've included in my talk things that will keep everybody interested. So, in summary, I'm going to talk about prions, DNA, prion diseases, and specifically the genetic forms of the disease as well. One of the things I've spent a lot of my life working on is discovering new genes that alter the risk of getting sporadic CJD. We've got some new research in this area that's not yet published, but is about to be published. And I'm going to try my best to explain how we've made these discoveries and what they mean for families. I'm going to talk also about the inherited prion diseases that are specifically genetic disorders, and talk somewhat about the special opportunities that occur in families. And I think the main new understanding of this research is about understanding the fundamentals of disease. And I want to try and get across the ways in which we're learning about sporadic CJD and prion diseases in this talk. So finally, I can't talk much about this, but I know people will be interested about our experimental treatments with PRN100 that I was prescribing for patients in London. And I have a single slide to give the latest update on that treatment. So to talk about genetics, perhaps people don't remember from being a newborn baby that the conversations around the cot or around us lying in our mother's arms were on the subject of genetics. Because of course the common conversation is how our appearance as a baby matches more distant people in the family, whether we got our grandmother's nose or our father's eyes. And the illustrations I put on this slide concern identical twins, which are perhaps the most striking example of how genetics controls things in our body. But of course, these similarities, the similarities between identical twins and the effects that are caused by genetics go way beyond the superficial, because genes determine, to some extent, which diseases we will get in life. And this is determined because genes are variable. We all have different DNA codes. Now, some of those changes in the DNA code are highly common in the population, very frequently different at particular points, whereas others are rare, some extremely rare. Some variations or mutations are found only in a single family, a single individual, and some of them can have really profound effects on one's life, as some of you listening know only too well. Diseases are also variable. in that some diseases have a very large genetic component, and some have a large environmental component, and some don't really have much of either. And at the moment, based on our understanding, we can only say that they're mainly determined by luck. So I'm going to talk about, I mean, each of these have different examples that fit into those descriptions in the prion diseases, and I'm going to go on to talk about that now. So there are different causes of prion diseases. The most common human prion disease, sporadic Creutzfeldt-Jakob disease, that I'm sure has probably touched the majority of people listening to this call. Nothing I'm going to say in this talk changes the conclusion that has been long held by researchers that principally this is bad luck. We don't have a satisfactory explanation of why one person gets sporadic Creutzfeldt-Jakob disease and not the next person. But this, of course, is different for different types of prion disease. So the inherited prion diseases, or genetic CJD, is predominantly caused by a single genetic variation that occurs in the prion protein gene. So a single genetic mutation determines all cases that get, all patients that develop inherited prion disease. And then when we come to the acquired prion diseases, those that are being transmitted, like variant CJD or iatrogenic CJD, in some ways, the environment is most responsible. It's exposure to prions that determines this disease, whether it's BSE contaminating the diet or medical or surgical procedures that have transmitted prions in the case of iatrogenic CJD. So for those people that are fresh, just to recap some more details about the main types of human prion disease. So the vast majority is sporadic CJD, which is a rapidly progressive dementia condition that affects people in later life, from middle age into old age, with a peak in the 60s. This also is associated with neurological features, the jerky muscle movements of myoclonus and the loss of balance of cerebellar ataxia. And sadly, most patients will die within only a few months of the onset of the disorder, although it is quite variable between people. And as I mentioned, it seems to occur randomly in populations, with a risk over a lifetime of approximately 1 in 5,000. Now that number may be a little bit of a surprise to some people listening, because we hear so often about the 1 in a million, but of course 1 in a million of the population is a per year number. And actually, if you multiply that up by the number of years you're likely to live, you don't get far from 1 in 5,000. The 1 in 5,000 comes from a very simple calculation, in fact, from England, where approximately 100 people die from sporadic CJD every year, and we have about 500,000 deaths in total. And that's how you get 1 in 5,000. So sporadic CJD, the big clue about the cause of sporadic CJD is that it doesn't run in families. So, and there are by definition, there are no known environmental exposures. Now, because the condition occurs at this 1 in 5,000 chance, there will be from time to time an observation that 2 people in a family get sporadic CJD. And indeed, a few families like that have been reported, but not beyond what would be expected just by chance. So sporadic CJD is a bad luck disease, but what does this mean? What does this mean in terms of molecules? So in the brain, we think the basic cause, the first thing that happened that determines somebody to get sporadic CJD, is a spontaneous change in the shape of prion protein in a brain cell. Or alternatively, you could frame it differently and think that maybe there's a mutation in the DNA code that makes the prion protein. So you have a brain cell perhaps that is manufacturing an abnormal prion protein that is predisposed to change to the abnormal prion form, but is then, that DNA change is not passed on in the family because it's only occurred in a single cell in the brain. Whether or not research will ever be able to identify whether these processes are really happening or not, I don't know. It certainly seems almost impossible to be able to characterize an event like that in a person once a disease has developed. But so you might say, well, why if it is just a bad luck disease, why am I spending much of my career trying to research genetics of CJD? And what I'm after is to try and find genetic variation that alters the risk of getting the disease. But from a family point of view, this might mean going from what would be a lifetime risk of 1 in 5,000 to an increased risk of 1 in 4,000. Now that of course isn't really meaningful. Nobody walks around worrying about having a 1 in 4,000 chance of getting a disease versus a 1 in 5,000. But the reason to be motivated for this is about tackling a problem. So, faced with problems in life, how do we go about it? We first want to try and understand what the problem is. So this is about trying to understand the molecular underpinning of a disease so that we can plan how we're going to tackle it. But I have to say, I research on genetics, I have to say something about the inherited prion disease that are predominantly genetic disorders, but these are incredibly varied brain diseases. Some of them look just exactly like CJD, aside from a family history of the disorder. But there are these quite distinct clinical pictures that can come up with the inherited or genetic form of the disease, such as the sleep problems and autonomic nervous system problems of fatal familial insomnia, the balance problems first and then only later difficulties with thinking in Gerstmann-Strauss-Scheinker syndrome, which progresses much more slowly than CJD over 4 or 5 years. And then less common even some insertional disorders in the prion protein gene that cause very, very slowly progressive problems with thinking that can go on over decades. And even some non-neurological presentations that can have as the first symptom diarrhea related to abnormalities of the gut motility that can last for many decades, caused by mutations that truncate the prion protein. So inherited prion diseases are caused by a mutation in the DNA code that makes the prion protein. Now we each have 2 copies of this code, and the mutation is found in only a single copy. And importantly, that determines the risk in families. So for a patient that has had the disease, the risk to their children is 50-50, because we each pass just one of our genes. Each gene is passed on just once to our child, and the other half of the genetic code comes from the other parent. So that means the risk at birth of passing on a mutation is 50-50. Now, because the inherited prion diseases can look like sporadic CJD, we offer a test to rule out this possibility in every patient that we see with apparently sporadic CJD, and in the vast majority of cases are able to give reassurance that there is no then increased risk to the family because we've eliminated an error in the DNA code. So in this disease type, genetic variation explains pretty much all the risk in a lifetime of whether you're going to get the disease or not, but not all of the risk. And the reason I say that is there are some gene mutations of the prion protein gene that confer a risk of disease that is almost guaranteed to happen. It may start early in, relatively early in adulthood, and that nobody ever carries the gene mutation into old age. But other genetic variants cause a much later onset of disease when other medical problems are more prevalent, and people may well die of something else before the disease develops. This age at which the condition starts is very variable, and we don't really have adequate explanations of why it starts at a particular age in one person and not the next. And this is a really active area of research. We know that genetic factors can explain some of it in some of the mutations, not very many of them, not in the CJD ones typically, but in, say, in the insertional mutations, there are strong effects of a variation in the prion protein gene called codon 129 in determining when the illness might start. So the reason we do genetic research in the inherited prion diseases is to be confident about which mutations actually cause disease, at what age do they typically cause a disease, what kind of disease that is, in order to counsel people and explain what they face in their family and help them make choices, whether that's about reproduction or about having a test to see if a mutation is being passed on. So, what I'm talking about here is the special opportunities of being at risk of inherited prion disease because of a diagnosis in a blood relative. And by saying special, I mean, some big decisions in life about whether to have a test to see if you as a healthy person have inherited a mutation from an affected relative, decide whether or not to have children, or to use reproductive technologies that might allow you to have a child that you know cannot possibly carry it, even if you do. And then potentially to become actively involved in clinical trials or studies with the aim of prediction of the disease better, or prevention of the disease if there is, if such trials are to be done, whereby a compound is given with the aim of preventing the disease starting at all. So I haven't covered much molecular biology, and in order to properly explain some of the research work I'm doing, there are some basics to understand, and I do apologize for this, and I don't want it to sound like a class at school, but there are some basics of cell biology and genetics that I'd like to talk about for a little while. So you had two other things to understand if you're going to understand the research we're doing. So this is a picture of a cell, a cartoon of a cell. Now, cells make up the structure of our body. We have billions of cells. They do all kinds of different muscle cells, brain cells do our thinking, and blood cells carry oxygen. Each of them are adapted to different functions, but they have some similarities too. They're all small. Typically a 1/10 or 100th of a millimeter across. They're surrounded by a lipid bilayer, so a fatty membrane that contains all of the structures inside the cell. And there are lots of these different structures that have different functions, even smaller than 100th of a millimeter across. But most importantly for this discussion, this structure, which is the nucleus. Okay, now the nucleus contains the genetic code that tells the cell how to build its proteins. Okay, now we're going to talk a bit more about that. So this is the structure of DNA, the double helix structure, and these different colored bars are the bases, the nucleotide bases of DNA, and the different colors represent different letters in the alphabet of the DNA code. Now there are only 4 letters in that code: A, G, T, and C, and the instructions here in this code tell the body how to make all of the hundreds of thousands of proteins that we have. So how does that happen? Well, and don't worry, there aren't many of these slides for those that get baffled by molecular biology. So the nucleus packages up small sequences of DNA code as RNA. It exports it from the nucleus, and then this machinery here then translates the code to make proteins. So how does it do that? Well, proteins are made up of amino acids that are little chemicals, and these are shown here by these orange dots. And we have 20 amino acids, and here's a chain of them that is forming a peptide or a protein. Okay, so a protein, when this folds up, it would be a mature protein. So for those of you who have been following closely, you'll see the DNA code has only 4 different letters. We have 20 different amino acids. So how does that work? How's that translated? Well, for each amino acid, you need 3 letters of the RNA code to tell the body which amino acid to add next onto the chain. So when I talk about a genetic variation, I'm talking about just one of these letters potentially being different. You can also have other funny kinds of variation, like the addition of extra letters or letters being deleted or being flipped around, but mostly you're talking about just one letter that's been exchanged for another letter, just as simple as that. And there are 3 billion letters in total. So you can imagine how trivial just a single gene mutation might seem to have such devastating potential consequences. So onto some more specifics. So on the left here, you have what we believe is the structure of the normal form of the prion protein, which is one of our body's 100,000 or so proteins. So this in the cell typically lives on the cell surface. It's attached to that lipid bilayer, the membrane. It has a structured domain that comprises 3 helices, 3 alpha helices. Sugars can be attached, and there is an unstructured domain that floats. Well, we don't know what structure it adopts in the real cellular situation, but I can't show its structure here. It's not been determined. But what about the prion process? So focus on this top panel here. What you need to imagine is this red block here is a prion, and that consists of multiple abnormal prion proteins which have a completely different shape. and they're all stacked up together to form a stable structure, which we're calling a seed, which the body really finds it hard to destroy. And that seed has a certain properties, the prion. It can bind that normal form. So that normal form is illustrated by one of these spheres. The seed can bind the normal form and cause it to unravel and take on this abnormal structure, and thereby grows the seed. And then as the seed grows, it can fragment to create more seeds. And that's a chain reaction, because the product of the reaction forms more substrate to make the reaction happen again. And this, I think, is partly why prion diseases are so devastating, because of this cycling reaction to manufacture more and more prions. Now, the reason I've got different shapes and colors here is just to illustrate the point that we think these different types, I've described some of the types of inherited prion diseases, some of the different clinical pictures that we see, we think are probably conferred by different shapes of the abnormal form, or strains, as we call them, that have different kinetic properties. So different parts of this pathway might work faster or slower, and might allow prions to grow faster or cause more damage in particular brain regions. So in this slide, I want to illustrate the DNA variations that we've now described in the prion protein gene. So here in the center here, this cartoon illustrates the sequence of the prion protein gene kind of laid out straight. And these different colors represent different sections of the prion protein gene. And you see now, we've now learned a huge amount, because we've been sequencing, learning the code of this in tens of thousands of patients and healthy people for, well, over 30 years now. And we've learned a huge amount about genetic variation that can be seen, and it has a wide range of different impacts. So at the top here, what I've called definite, these changes in the genetic code, we are absolutely confident lead to inherited prion disease. So we're sure that a disease that is passed on from one generation to the next can be caused by each of these genetic variations. But sometimes we just make an observation of a genetic change in one person with CJD. And what we don't know, they may not have it running in the family, and we may never have seen it anywhere else in any healthy person or any sick person. And those, well, we're a bit suspicious. Maybe it causes the disease, but we haven't really got enough evidence. And we bunch these together here. Now, other variations we've found just seem to be just as common in the healthy population as they are in patients, appear to do nothing at all. And then we've also found some variants, this one at position 129 is particularly important, that seem to alter the risk of getting disease, and in some particular circumstances are protective, or even close to completely protective. So another way of putting that is that there's a spectrum of genetic variation that goes all the way from variants that we've found that are going to pretty much guarantee, sadly, that you will develop the disease in your lifetime if you live long enough, and others that may confer complete or almost complete protection from developing the disease, and then the whole range in between, including those that massively increase your risk of getting CJD from 1 in 5,000 to maybe 60 to 70% chance of developing the disease. But we also might know from families and from testing that there are some individuals who've lived into a perfectly healthy old age that carry these genetic variants that haven't developed signs of the disease. And so genetic research at the prion protein gene aims to build the evidence profile for every genetic variation we found, so that when we find a new patient with a genetic variant that's been seen before, we can properly counsel those people about what to expect. And that's really important, that we give as good information as possible. I talked about some of the other effects that genetic variation can have and the importance of position 129. So this chart shows the everyday functions of patients with sporadic CJD and the typical linear rates of decline in those functions over time. So here, days on this axis here. And you can see there are 3 different patterns that we tend to see. And those 3 different patterns are found in 3 different genetic types that we see at position 129, with the very fastest patients with sporadic CJD having only methionine type amino acid at that position, and the slowest having 2 different types, methionine and valine, and in between being those with just valine. And this factor is crucial in how we plan to do clinical trials in sporadic CJD because of these profound effects. Codon 129 also appears to have some really strong influences on the strains that I referred to, the different shapes of prion that can form in disease. So just to take a little pause there, I've kind of introduced the causes and main symptoms of different types of prion diseases and talked about the fundamental differences in what causes the disease, whether it's bad luck, whether it's purely genetics really, or whether it's about specific environmental exposures. And I've tried to explain some of the underpinning molecular mechanisms, particularly talking about the wide range of different genetic variants we've seen at the prion protein gene, and how some of those are common and can alter how the disease manifests and the risks of it, and some of them are extremely rare, but may be very, very significant for a particular family's risk of disease in their lifetime. So what I would like to go on to do is to try and explain something about well, I mean, it would not be humble of me to describe it as cutting edge research, but some of the research that is just about to be published and new discoveries in terms of genes that may alter the risk of CJD. So this research technique I've been using with collaborators is called Genomewide association study. So one of the big research projects I've had in my life is this technique, and I want to explain a little bit about how this works. So the idea here is to discover new risk factors for CJD. How do we go about it? The main aim is to, or the main objective is to collect together DNA samples from as many patients who've sadly had this disease as possible. And in order to have success with a project like this, you need many thousands. And so I think it will be immediately obvious to people for such a rare disease how challenging that is. You also, in order to do this, need access to similar or even larger populations of healthy individuals. What you do then is you take these panels of DNA all plated out in the laboratory, and you run them through a machine that uses arrays like I've illustrated here from the company Illumina. And using these arrays, you can measure at the same time hundreds and thousands of these common genetic variations that define major differences between individuals in a population. After a quality control of those measurements, you can fill in all the gaps. So in total, we have many millions, 10, 20 million, that kind of order of common genetic variants across the whole of our genome. And we can use computational techniques to fill in the genotypes, the variants at all of those missing sites. And then essentially you're comparing. You're saying, out of those many millions of genetic variants that you've described in these 2 large populations, are any of them more common in patients with CJD than in the healthy population? It's as simple as that. So the main determinant of success would be the ability for everyone in the world that is concerned with CJD, that is collecting DNA samples from patients, to club together with the purpose of making new discoveries. And one thing, one important purpose of a talk like this is for me to say thank you for everybody doing that. And it's perhaps useful for you to know that experts around the world are very keen to collaborate and share resources, because they know that only through sharing will we make any discoveries at all. And so pretty much the research I'm presenting here represents the whole world getting together to share their resource. Actually, there's an important caveat I must say on this, that until recently, the main priority or the main focus of genome-wide association study has been European ancestries population. So you're talking white European ancestry. And that is, I think, primarily because it's these countries that have had well-developed healthcare systems that have allowed the collection of large numbers of samples. And a key priority going forward, this is worth repeating, is to understand genetic risk factors in other populations. So when I say the whole world, that's not, of course, true, is it? But there are no large collections, for example, of African patients with CJD that would allow a study like this. In any case, what I want to show in this slide is that we've collected over 4,000, for the discovery stage, over 4,000 cases of patients that have sadly died from sporadic CJD. And there are two phases to the study. That's a discovery phase and then a check phase where we look again and see whether in a completely new sample we find the same thing again. And again, there was a great collaboration from experts in these countries, providing a new set of cases and controls. Now, one of the major risks of doing a study like this is that your cases are not really matched very well to your controls. You know, it's the same problem in epidemiology, and you may have come across this problem. If you don't compare like with like, you will get a spurious result. So this is a complex statistical technique I'm showing in this slide, but the point I wanted to make is we're very careful in the way we've done this to try and make sure that we haven't got a spurious finding because our controls were not well matched to the cases. And this is a data summary technique called principal component analysis, where we map on in the colored dots all the patients against in the gray squares all the healthy controls. And you see overall genetically, the cases are the same as the controls. And that was important for us because that means the study is not strongly biased. Now, I'm not going to go through the technical details of how you actually do the analysis, but these are the main results. Okay, now this is called a Manhattan plot because these towers, these different colored towers that you see, look like the Manhattan skyline. Now, I'll just want to explain what this main result shows. So each of these different colored blocks show different chromosomes. These are different packages of DNA. And here is the statistical strength of our finding. And this is the mathematical term minus log p. Now, this blue line represents success. So at that blue line, we can start to be pretty confident we've got a hit. This is a new discovery. And one of the really important things to find, which is not a new discovery, but it was an important proof of principle for me, is that we could actually find the prion protein gene using this technique. If we couldn't find the prion protein gene, that would be a big problem because we already know that there are strong genetic effects there. And so I was delighted to find when I looked at the results that there was this big skyscraper right over the prion protein gene on chromosome 20. And so I was strongly reassured by that. But also the next thing I looked for, are there any new skyscrapers that we didn't know of before? And there were two. There's one of this gene, Gal3 ST1, and one at another gene, STX6, this one on chromosome 1, this one, Gal3 ST1 on chromosome 22. There were two other genes that were nearly there, and we went on to test in the replication study, but couldn't confirm, so I'm not going to talk about those anymore. So here's a table of results. I apologize for this being quite technical. There are a couple of points I want to make. These are our 3 hit genetic variants. This one already know. This one reflects the codon 129 variant I've already described. I wanted to just illustrate to make the point about what this means for families. That the risk variant, we found 45% of DNA strands from CJD patients, 41% in the healthy population. So this in itself does not determine whether you're getting CJD or not. Okay, now the chances of that, were the chances of finding this spuriously, were less than 1 in a billion. So this was a really strong and confident finding, but it's only a modest effect. So you got to remember that this is not like discovering a new gene for inherited prion disease. This is subtly changing the risk of CJD, and similarly for the variant of Gal3 ST1. So just to summarize this genome-wide association study work, we're confident that we've now got 3 genetic risk factors for sporadic CJD. And in the remainder of the talk, before I finish on PRM 100, I just want to say a little bit about what we're doing to understand what this means to understand the disease better. And so how do these genetic effects work? So, the first thing that you want to do when you found a genetic region that appears to confer increased risk of CJD is to ask, well, actually, which protein is made at this locus that is really conferring the altered risk? And this was relatively easy at both of the regions we discovered. So at STX6, that encodes Syntaxin 6, which is a protein. And what we found is the message that goes on to make Syntaxin 6 is increased in crucial brain regions. The caudate and the putamen that are typically abnormal on MRI scans showed increased amounts of this message being made when you had the risk variant. So we think more Syntaxin 6 means more risk of sporadic CJD. So maybe if you reduce Syntaxin 6, you would have less risk. And that's one hypothesis. But what about Syntaxin 6? So we used antibodies to look under the microscope in brain tissue about where it is. And you see these brown blobs in this cell here, the Purkinje cell, showing that there are particular structures inside the cell, particular vesicles, we think, that have this protein on the surface. What does this potentially mean? Well, here's another blow up of a cell. I'm not expecting you to remember all the details from the previous slides. But needless to say, what we know in prion disease is these abnormal prion seeds are moved around the cell a lot. They may form on the cell surface, but then they come in, and sometimes they can be transported to structures that might destroy them, or they can go back to the cell surface and could grow further. Or maybe they will meet new prion protein that's been made in the cell and convert it to an abnormal form. So what we think Syntaxin 6 is doing is favoring a process through which prions either can grow faster or can be more toxic by changing the way in which these vesicles or these small packages, structures inside the cell are being moved around. The other thing we discovered about Syntaxin 6 is it's also a risk factor for another disease, progressive supranuclear palsy, which previously wasn't particularly related to CJD. It's an uncommon neurodegenerative disease that looks a bit like Parkinson's disease without so much tremor, but is linked to the protein tau, which is misfolded, aggregated, and deposited in brain tissue. So we found a genetic variant that appears to increase risk of two related diseases, and there must be a shared mechanism here, which is intriguing, I think, and helps further research in this area. But what about Gal3 ST1? This encodes an enzyme, and we were lucky in that the genetic risk variant appears to alter the primary structure, the protein structure of that enzyme. And we know already that this enzyme is involved in the synthesis of brain fats, brain lipids. Now, the enzyme encodes this step, which is the conversion of this lipid galactocerebroside to sulfatide. This is the structure of sulfatide. It's the major component of the myelin sheath, the cable that wraps around axons in the brain. Now, there are various strands of research that link prion protein to myelin structures. And so this, again, is an active area of research of how this lipid component can alter the risk of getting CJD. So my conclusions. A big thank you to everybody that's contributed DNA samples. And I don't just mean the experts that have shared their collections. I mean all those thousands of individuals that signed consent forms to donate their DNA after gene tests were done around the world. And working together across the countries I showed, we have discovered, I think, evidence now for 2 new genes that alter the risk of CJD. Now, these are small alterations in risk, not like a prion protein gene mutation, but we're confident in the changes. I don't think there are specific implications for families. This doesn't change the fact that sporadic CJD risk is not conferred within a family. Just because you're a relative of a patient who died of sporadic CJD, you're not at a marked increase in risk of developing the disease in your lifetime. But it does, I think, give us some avenues to help understand sporadic CJD better. And I don't want to say that immediately that these are drug targets and that we're going to develop a treatment this way. But the whole idea, the grand plan of our research is that we thoroughly understand a disease and then we can design treatments. I mean, it's a step and it's a piece of the puzzle in trying to get a complete picture of how the disease is caused. Now, there is a future for genetic research too. So one of the things we're actively doing at the moment is looking at genetic determinants of how fast the disease progresses. We want to try and find a gene that slows it down, because that also potentially could be an interesting target to consider whether it might be amenable to drug treatment. But I just want to finish on PRM 100 because I know there are probably people listening that will be interested, and it's good to be on the subject of drug targets because of course our main drug target is the prion protein, and PRM 100 is a manufactured antibody that we've made that binds the normal form of the prion protein. Now, if you go back to the prion mechanism, the idea behind this treatment is if you bind onto the normal form of the prion protein, you remove the fuel for the production of more prions, okay? Because once the prion protein is bound to an antibody, it can't participate in the growth of the prion seeds. And the idea is that the disease will just burn out of its own accord. So this is a long history to how this experimental treatment was developed. But to be brief with where we're at with this, we started treating patients with manufactured PRM 100 in October 2018, following a hearing at the Court of Protection that such treatment was lawful and in the best interests of a patient that could not give capacity to decide. Now, in total now, we've now finished treatment and there is no antibody remaining to treat new patients. In total, we treated 6 patients and we treated them for a variable length of time, depending on how long they survived. And there are a number of positive findings. We didn't see any major drug reactions, no evidence of side effects. This appeared to be a harmless treatment in those that we did, and including, in some cases, many months of treatment at the high doses. In our plan, we developed a strategy to be able to deliver this directly into the brain if we needed to, because the brain is surrounded by a membrane that stops large molecules like antibodies crossing from the blood into the brain. And we were giving this antibody via a drip infusion into a blood vessel. Now, another encouraging finding was that we hit target levels of drug in the spinal fluid, and we did not need to progress to giving this directly into the brain that would have required neurosurgery. Now, of course, what everyone wants to know is, was there a clinical response? And I'm sad to report that all the patients that we treated have now died, and nobody was cured from CJD. But I think that was only the very most unrealistic and optimistic expectations that our first attempt to treat in this way would result in such a dramatic clinical response. I think it's fair to say it's the variation in the rate at which patients decline is too great to draw conclusions from only treating 6 people. Now, I wish I could say more about exactly where we're at the moment, but hopefully it suffices to say that it is the #1 priority for us to further develop this treatment. And we're actively involved in developing the next steps, which I hope to be able to share with you in the not too distant future. We will, of course, publicize any developments as soon as we can. So that's our update. That leads me to thank people. This is the National Prion Clinic team. Of course, sadly, we can't meet like this anymore, can we? We're all far too close there in that photo, so that's an old one. New members of staff kind of then pasted on at the side, but we're a happy team. I also need to thank those scientists, particularly Azi Khalili Shirazi, that have been involved in developing PRM 100 at the Institute of Prion Diseases. Here we are in Fitzrovia. And a lot of support came from a peer-led charitable campaign, the Cure CJD campaign, that provided a lot of funding for this purpose. So a huge thanks to them. And finally, I hope I haven't missed anyone off here. All the collaborators that contributed DNA samples for the Genomewide Association study, I really, really appreciate being able to work together in this way. Thank you so much for your attention.
All right, well, we'd like to thank everyone for hanging in there through those technical difficulties to hear Professor Mead's presentation. And we especially thank him because it is 1 a.m. where he is right now. So thank you for staying up with us. Now to review some of the questions. If we do not get to your question, please feel free to call us or email us at the help email box. So for the first question, if someone was tested for familial CJD in 2008, have the testing protocols changed to where that person would benefit from being retested?
Hi, thanks for the question. I don't think there would be any benefit in retesting, no. The mutations are all found in a single gene, the prion protein gene, and the protocols have been in place since the early 1990s that are comprehensive in picking up any variation in that gene. So that would be a definitive result, and I don't see any benefit in repeating that. I notice the question goes on to ask about whether or not screening through Ancestry or 23andMe would identify mutations. And well, each commercial provider has their own mutations that they announce they will screen for, and that should have been clear up front before having any DNA testing through a company. My understanding is that those companies offer a limited number of clinically relevant gene mutation testing, and that doesn't include the prion protein gene. Of course, you know, things may well change re commercial testing at any point in time, and you know, so that's the current situation as I understand it. And personally, in our clinic, we have never met yet a patient that has uncovered that there is a risk like this in the family through some kind of private commercial testing. I certainly expect that will happen at some point in the not too distant future, but so far it's not happened.
Okay, so next question. The writer believes that their father had sporadic CJD. Should they get genetic testing done?
Yeah, sure. This question does come up from time to time, and if I understand the question correctly, what the questioner, the situation that the questioner is in is that their parent has died, and there are no samples remaining to test the parent. So that would be the first choice if it had been possible to try and retrieve material. This could be a blood sample, it could be a tissue sample if there was a post-mortem examination done and brain tissue was stored. The most efficient way to proceed to rule out a genetic risk in the family is to test somebody that has died from the condition, because that then rules out the possibility of inherited prion disease, genetic CJD entirely. So assuming that no samples are available at all, my practice is to provide reassurance to these people based on scrutiny of the clinical case. So in other words, I review the clinical investigation features, the history, the examination, get a detailed family history, and if all of that investigation points to a typical case of sporadic CJD, then I don't advocate any genetic testing. The situation for us has never arisen whereby we've done a blind genetic test. And what I mean by a blind genetic test is that there are hints in the family story or investigation that there might be a genetic prion disease, but there are no samples remaining from anyone that's been affected in the family. Now, in that circumstance, potentially you could screen perfectly healthy blood relatives to see whether or not there was a gene mutation or not. Of course, the only thing you can really find there is, if you get a positive result, is bad news for that individual. So far, We have never done that. We haven't had a policy of accepting blind testing where there is no gene mutation known in a healthy person. I, however, don't see that as an insurmountable thing. I could be persuaded that that would be a reasonable thing to do in some circumstances, just that we never have done it. So I'm sorry this was a rather roundabout answer to what seems a straightforward question. So just to summarize, my suggestion is if the father, in this case, had a typical history of sporadic CJD from the point of view of the story, the investigations, then the chances of having a mutation are extremely low, and I would not advise testing.
Okay, so next question is, can a father pass it to their child? And while the child is in the womb, pass it to the mother through the placenta? Yeah,
sure. Well, of course, from the point of view of the DNA that you inherit, half comes from father, half from mother. So if father is a carrier or died of inherited prion disease, genetic CJD, then of course that could be passed on to a child. And the risks of the child inheriting the gene mutation are 50-50. Now, the question implies, though, are there other ways in which the disease might be transmitted? I think that's the implication of the question. In other words, you know, for example, if the mum had sporadic CJD whilst pregnant, but no gene mutation, could the disease be passed on in that way? In other words, could prions cross the placenta and enter the baby and cause a risk for that baby subsequently to get CJD, even though there's no gene mutation being passed on in the family? And so as far as we know, and similarly, you could imagine other mechanisms in families by which it might be transmitted from person to person without a gene mutation. But we don't have evidence that that has happened. It's certainly theoretically possible, but there are no proven cases. So the counselling I offer people in families is that for the time being, at least, we believe there is no risk of this happening.
Okay, and I think the other part of that question was, if the father passes the gene to the child, can the child in utero? Oh, I see. Yes. To the mother?
No, that's a good. That's certainly an aspect of the question that I missed. Apologies for that. Yeah. Well, again, I think it's the same answer that I gave. Theoretically, this is possible. Okay, because we know that baby cells do cross into mum, and baby proteins will cross into mum when the baby is growing in utero. So theoretically, there could be a risk of passing on prions in that way. Certainly, it is not a proven thing to have happened. So, you know, in order for it to be proven in epidemiological terms, it would require, you know, a number of these cases to be apparent in families that are passing on gene mutations. So there would have to be a number of married-in mums that developed CJD or another form of prion disease, having had a baby that carries a gene mutation or subsequently went on to get the disease. Now, there is one such case in a Danish family, but that's only one. And as I mentioned when I was talking about 2 cases of CJD in one family, from time to time, these things will just happen by chance. You know, if you study enough families, there are enough married-in people that eventually one of those married-in people will unfortunately get CJD. So as it stands, there aren't enough cases like that to be confident that it can happen at all. I'm sorry, that's not an absolutely definitive response, but that's the current state of evidence, that we think basically this is not something that is definitively happening, but it's something we're alert to and we keep an eye on.
Okay, is there any way for people from the US to be part of this study you're doing? Are you interested in familial CJD families?
Yeah, sure. So absolutely, but perhaps I wasn't clear enough in my talk. There are the US, from the point of view of the Sporadic CJD Genome-Wide Association study, there are thousands of contributors, and largely these have come through the Case Western National CJD surveillance outfit, of which Brian is currently the director. Michael Geschwin's team in San Francisco also sent a number of samples to us. So, both of those teams have made contributions, very significant contributions. So there are definitely routes. From the point of view of familial CJD, yes, I am particularly interested in DNA from people that have or are at risk of familial CJD for these kind of studies too. I mean, at the moment, we think the age at which this condition starts in people is determined by the mutation type, and some of the mutations are controlled by the variation in the Prion Protein gene. But other than that, we know of little else. And there's some evidence that there's unlikely to be other strong genetic factors. But the only way to find for sure is to develop large samples where you can test this definitively by actually looking for them. And so yes, for certain, I am interested in DNA from familial CJD. And the simplest way for that to get to me is through leading research clinicians in the US. So you're talking through Brian Appleby or Michael Geschwin's contribution are two that have already expressed an interest in working with me with DNA samples from people with familial CJD. So that would be the most straightforward route, I think, if there's a connection there.
And if that writer was referring to the PRN 100 study, your answer would be that that's not in progress currently.
Correct, Toby, thank you. Yes, with respect to PRN 100, there is no option for anyone in the world at the moment because there is no manufactured antibody that we can give. So, eligibility when we have a way forward for the PRN 100 treatment will depend very much on exactly what that way forward is. For example, it could comprise clinical trials in multiple countries. That's one possibility, in which case there might be all kinds of routes to access the potential treatment down the line. But this is not determined yet.
Okay, next question. Are there any unique, potentially confounding variables that differ by geographic location that need to be controlled for when aggregating so much of this data from the US?
Yeah, that's a really good question. And I did somewhat gloss over some of the technical details and the statistical methods used to control for confounding variables, the main one being genetic ancestry. So that there's such a rich history to populations that correlates strongly with geography, that this could be a major confounder. And we did in fact use 10 axes or parameters derived from data summary techniques that I showed on a single slide, this principal components axis, principal components analysis. We used the first 10 dimensions of this data summary technique to try and control for genetic ancestry, which would correlate very strongly with geographical location. So we did control for that. But there are other things as well, though, that would be much harder to control for. And one of the things that worried me a little was the different ways in which CJD is diagnosed in the world and ascertained by different surveillance systems, in that you might get different types of CJD with different clinical histories based on how it's picked up. It didn't seem to be a major confounding effect in the end, in that we didn't see a great deal of heterogeneity in the associations that we found. In other words, the two hit genes that we got, the STX6 and the Gal3 ST1, were hits across the world in every large country, which was quite a useful finding, I think.
Okay, biochemical evidence suggests that cellular cofactors are involved in prion replication, including lipids.
Yeah.
And do you think that...
Gal3 ST1.
Sorry, I just lost it. Yeah,
no,
right.
I thought you were stuck over the gene symbol there, Debbie.
No, I lost my cursor, sorry.
Not at all. Absolutely, Jason, for sure, yes. This is something I would love to look into. So it does cross my mind. Could it be that sulfatide or its premetabolites like galactosylceramide are cofactors in prion replication, and therefore the concentration of these modified lipids at crucial sites could determine the speed of prion replication? I think that's an intriguing hypothesis. So, it may be that you might argue then that the risk variant at Gal3 ST1 maybe increases the concentration of cofactors that promote prion replication. Yeah. So I agree. Yeah. That would have to be one of the leading hypotheses to follow up on.
Okay, and if you carry the genetic version, does that mean that that person will die from it if they live long enough?
Well, yeah, that isn't that just the crucial question, and unfortunately it's complicated. It really depends exactly what is the genetic mutation that is carried. So just to make that more concrete, with an insertional mutation in the prion protein gene, with an extra 6 octopeptide repeats, which is a particular mutation type, then I've never seen anyone live beyond the age of 50 without showing signs of that disease. So for that particular genetic variation, most definitely, if you live to 50, you will develop signs of it, and patients will die of this before their early 50s. But it's not the same for others. So if you look at some of the particularly common ones, E200K, D178N, for both of those variants, I've met people in healthy old age that I know carry it in their so people that have lived to their 80s. And we know from family trees there are individuals that must have passed it on because they transmitted the mutation to somebody that we know carries it, and yet they live to healthy old age. So for those, no. I don't think you can say you will definitely get it, even if you live into a healthy, extreme old age. So it is complicated, and this is one of the crucial aspects to have a one-to-one discussion with an expert physician about, who can look into exactly which genetic mutation is carried, and then show you some charts and some, go through the latest data that has been aggregated over the last 20 or 30 years to have some confidence in what the chances are by which particular age. So not a straightforward answer, but so it depends.
But a lot more is known about penetrance and the differences among the different genetic mutations than was ever known before.
Absolutely, Debbie. Yeah, that's right. So we share our data. This is a rare disease. There's a limited number of expert physicians and other interested people around the world, and we're all convinced that the way to get the clearest data and answers to questions like this is for us to club together, share this information where we can, where we have permission to share it, so then we can try and be as definitive as possible. I mean, the problem, one of the big difficulties with inherited prion disease, familial CJD, is just how variable the age at which it starts is from one person to another. It is unfortunately at the moment quite unpredictable in when it's going to start. And I would love to be able to find ways to help people that really want to know more about when this might start. And there's different ways to do that. One is to try and look at factors that might determine the age at which it starts, like genetic factors. I described that a little. And the other potential avenue to this is to try and develop markers that predict being close to when it starts. In other words, to detect signs of this brewing or developing, the infection developing in an asymptomatic way before the symptoms begin, maybe a year or two later. And we are starting to make some progress in that respect, but it's not at the stage at which we have some kind of test that we can use that will help foretell the start of the disorder.
Okay, so I think running underneath the undercurrent of a lot of the questions all of us are asking tonight is, what's the best way for us to support research progress?
Yeah. Well, I think what the genetic studies I've talked about this evening rely on are giving permission to use DNA taken from a blood sample or a tissue sample for subsequent genetic research. So this might be a consent form that somebody is presented during their loved one's illness to give permission for genetic analysis to be done. It may be a consent form to ask for permission to use tissues for research after a post-mortem examination is done. So, it's hard to talk about these things whilst a family is in crisis, and there are more important issues for the doctor and the family to talk about. But if you could spare a few moments to consider permission for genetic research and for post-mortem to be done, that is how we accrue these collections of samples that are necessary to have the statistical power to make some discoveries.
Okay. Well, I thank you, Simon, for staying up late with us. We really appreciate it and the benefit of your knowledge.
Pleasure.
And
thank you
yeah.
for listening. I really appreciate you coming along.
Good. And for anyone who feels their question may not have been covered adequately, help@cjdfoundation.org or 800-659-1991. Thank you very much, and we wish you all a good night.
Good night, everyone.